Good afternoon. I'm Tom de Swaef, and I work at ILVO in Belgium, in Flanders. And you probably this slide will probably remind you on the last summers we had on your lawn, where your grass looked a little bit like this. And indeed, climate change is already happening, producing droughts, and these droughts have an impact on our lives, on our lawns, but also on agricultural production. And therefore, we need to adapt to this change in climate and adaptation strategies in agriculture. We need to do this in a systemic way, and at ILVO we also invest in a systemic approach to adapt to the climate change. And we do this on a couple of levels. First of all, sustainable soil management, because a proper soil management allows the soil to absorb more water, to retain more water, and just delay the events of drought stress. We also want to introduce new varieties which have a better tolerance to drought. We want to introduce new crops which happen to have an intrinsic better drought tolerance, such as sorghum or quinoa or soybean. We want to study new cropping systems, so cropping systems that are more resilient to drought stress, and I think of mulching techniques, but also agroforestry has potential in this domain. And then finally, also if irrigation is needed in some conditions, we better do it in an optimal way. But for today, I will focus on the aspect of breeding for drought tolerance. And you should know that at breeding, and especially when we talk about grasses, because the topic will be on grasses, well, there are some traits which are very important in the breeding. And for the main trait, and that's in every crop like that, that's yield. So yield should be high. In grasses, you have a specific trait called persistency, and that's actually the fact that your crop is persistent after winter, because grass is typically a multi-year crop, so it's not an annual crop. And then persistency after winter, for instance, is very important, but also persistency during drought periods. Disease resistance is very important. Also, the quality of the grass, because grasses are used for forage production, and the quality determines how efficient the ruminants, for instance, the cows, will digest this grass, and it will have an effect on the methane emissions, for instance, and also on the production level of the cows. And from a breeding perspective, it's also very important to have a high seed production. So you want to produce seeds because you want to sell these seeds on the market. Now, these are all traits which have been in a breeding for a long time, but now in recent years, drought tolerance is an additional trait at which also our breeders at ILVO are looking. Now, breeding is a very slow process. Just to give you an idea, this is a typical breeding cycle. And of course, we do this cycle, we restart this cycle every year, and we let several programs run next to each other. But we start in the first 3 years by screening over 10,000 of plants, individual grass plants. Therefrom, we select interesting individuals, about 750, and these are then planted in rows in the next years. And these rows are then actually selected for disease tolerance. And then from these 750 clones, we do about 150 crosses. So we put different genotypes together, we produce seed from the crosses, and we then, in year 6 until 8, we sow these of this offspring into production fields, and then we check in a couple of years which are the most promising candidate varieties. So we select about 5 candidate varieties in year 9 already. We're already in year 9 in this stage. Then from these, we typically send the two best candidates we send for application in the to get accepted on the cultivar lists, and from that one, then typically we get one new variety, and then by that time we're in year 14. So it takes a long time, and you can imagine that it's very important in the first phase when you have 10,000 plants that you get a very good idea on the performance of these plants. So you really want to do a good job at this stage, and this is what this presentation is about. So in this presentation, we will talk about how a UAV, so a drone, can help us in the screening of drought stress tolerance in grasses. And this work has recently been published in remote sensing. So, and it already goes back from 2013. And at that time, we worked together with Ghent University with Professor Wouter. Now, in this trial, we have rain-out shelters, as you see them in the slide. We have 3 rain-out shelters next to each other, and so the trial is actually replicated 3 times. And in this trial, we all have individual plants, so clones, and there are 3 clones select for in every rain-out shelter. There's one clone, so the one clone is replicated 3 times. And we have 3 species, actually 2 species, tall fescue, which is Festuca arundinacea, and the second species is Lolium perenne. And from Lolium perenne, we have a diploid and a tetraploid group of plants. And in each of these 3 groups, we have about 150 genotypes that we monitored. And in this research, we had 2 main research questions. First of all, can the drone replace or complement the breeder? So the breeder typically will go to the field and he will visually go and say this is a good plant, this is a bad plant. He will address scores from 1 to 9 to each and every plant. And the second research question is, can we, within this pool of plants, can we actually find plants that have a higher tolerance to drought compared to others? So actually within each of the 3 groups, can we find better plants with better drought tolerance? So this is a bit an overview of our experimental layout. So what we see here, here we see the beginning of the season. So this is the 1st of April, and then we have a period in which the grass is growing very slowly. And then here we have a first mowing event. This is still outside of the experiment I'm talking about. And then we have a second mowing event. And this is actually the start of our experiment. And you also see that at that time we installed soil moisture sensors, which were required to monitor the decrease in water availability in the soil. And what we did, like these two green bars, these represent the timing of the rain-out shelter application. So during these time frames, we put the rain-out shelters over the field to actually introduce the drought in the crop. And these lines, this black line here you see, and then the other ones here, these represent the cumulative water deficit that we actually realized. So the black line is the cumulative water deficit we realized, and that's a typical metric to define how which kind of level of drought stress is imposed from the atmospheric level. And you see this line, actually this green area, is the most extreme example we have encountered in the last 40 years in Flanders. And the green one here, the lighter green one line, that's actually the line that corresponds to 75 percentile of cumulative water deficit over the last 40 years. And so the black line is the one we actually realized by using the rain-out shelter, and the dashed line is the line we would have had if we didn't use rain-out shelters. So just to show you that we actually can introduce drought stress by using mobile rain-out shelters. Now you see here the M indicates a mowing event. Here you have another mowing event, and here there's the last mowing event. And in between, so our experimental period ranges from this one, from the second mowing event until the fourth mowing event. And then you see we have 5 timings, 5 flights. So each arrow corresponds to a flight. And then at time 2, time 4, and time 5, also our breeder had scored the plants. And what is very, very typical for grasses and also very difficult for screening grasses is the fact that mowing times can come in right in the middle of your treatment. So a drought, this is a progressive drought treatment. And then this is a slow process. And then it happens that in the meantime you need to mow your plants. So this is something which of course is not the case like when you do wheat or maize or something like that, but this is the case in forage grasses. So that's something we need to take into account. Now I talked about the drone flights. We have 5 drone flights, and in each of these drone flights we used a very simple RGB camera. It's a Sony Alpha 6000, and this drone camera, this simple camera produced a lot of images, and these were done with the stitching software. They were connected to each other, and then you get this nice overview of the field. So you clearly see here the 3 subplots with the which are actually representing the 3 rainout shelters and also the 3 replications. And from these RGB, that's very important. You have a red channel, a green channel, and a blue channel. And from these 3 separate channels, so these actually give the intensity of reflectance in these wavelengths. You can calculate certain indices which refer to greenness most of all. Now, besides this RGB camera, at every time you also did a flight with a thermal camera, and the thermal camera was a FLIR A320. Now, why a thermal camera? Well, plants take up water, of course, but actually they lose most of their water. About 90 to 95% of the water they actually take up, they lose through their leaves, through their stomates, and this has a very important function because this allows these plants to decrease their leaf temperature, to get rid of excess heat. It's like a person who is sweating. He wants to get rid of this excess heat, and he does that by transpiring. So by evaporating water at the surface. So that's exactly how the same as how humans do it. But the very interesting thing here is that we can, if we monitor the temperature, we can get an idea of the amount of water they lose. And here there's a very important physiological mechanism, and it's called the stomatal conductance. So at the leaf level, you have this very small pores called the stomates, and these can be actively regulated by the plant. And if a plant senses drought stress, if it says, okay, I need to reduce my temperature, but I simply don't have enough water, then it will close its stomates, and then the temperature will rise. And that's exactly what we see here. So we see the image, it's a false color image, but you see here that there's a clear variation in temperature over the field. And what we see here, for instance, is this is a tall fescue part of the plot. The second replicate of tall fescue is here, and the third replicate is here. So you clearly see that the tall fescue subplot, in fact, shows much cooler, bluer, so much cooler temperatures compared to the rest of the plots. So this means that they continue to transpire as if nothing is really happening, as if there's no drought stress, as if the water is still available. So this is some very interesting insights. And of course, this is something that we cannot see with our eye. You can say, yeah, for the RGB camera, this is actually also what we see, but this is a wavelength we cannot see. So this is very interesting additional information. Now, I already explained that from the red, green, and blue channel of the RGB camera, you can calculate indices. So that's when we combine these channels in certain mathematical formula, very simple or a more complex formula, we get indices that more or less correspond to what we visually would say, okay, this is a good plant, this is a bad plant. And you see here the asterisks. These are indicating the indices that, in our case, show the very good correspondence to what the breeder said at these particular times. So you see that you can actually get some very high correlations, about 0.9 of correlation. So very high correlations just by a very simple camera, a very simple RGB camera. Now, what we also did was transform these RGB channels into other color spaces. So you have the hue saturation value color space or the CLAP color space, and these are actually additional mathematical transformations. And we see that, for instance, the hue value, the H in this graph, also shows to be very promising, and the same holds for the ND lab index. And these are thus not directly from the R, G, and B channels, but actually coming from from a transformation to another color channel. And CLOV is another one, and then you also see that the ND love is a very good proxy for what the breeder actually sees. Apart from so these are all indices which are derived from the RGB camera, whether or not it has been transformed to another color space. And then the last two, the delta T and the CWSI, these are indices that are inferred from the thermal camera. So these are actually a little bit these are from the other camera. And what we see here is actually that the correlation with the breeder is a lot lower in comparison with the RGB derived indices. So you can take this in two ways. So you can say, okay, yeah, this thermal derived images are not very interesting because they don't correspond to our breeder. But you can also look at it from another perspective. Maybe they tell us something additional to what the breeder sees, because actually these are things we cannot see, or not immediately. We can see this with a delay, let's say. So this is a very interesting finding. And then, which is very important in a breeding trial, of course, is the traits. Are these actually what we see on the plant, the values we give to the plant, the indices we give to the plant, to that specific plant, is that due to the genetic conformation of the plant, or is it just random noise we pick up? That's very important, because you really want to capture the genetic diversity and not the random diversity. And for this, we can calculate the heritability of the traits, and the heritability in this case is actually the part of the total variation we monitor in our experiment. What the proportion of that total variation can be attributed to the fact that they are different genotypes. So this is a statistical procedure, and then we see that actually the selected. So these are all the indices which were marked with an asterisk in the previous slide. So these come back, and then you see that there's actually some of them at least show a very good heritability in comparison to the score. So here we see, for instance, the H performs very well. The H is the hue derived index, and also the MGRVI and the VARI are very good, are very promising in this case. We also see that the CWSI is not very good performing in terms of very well performing in terms of heritability. So this is also important to take into account. Now, let's select these best indices from both whether they correspond very well with the breeder and they show a very high heritability. So then we take the score in the top line of this graph is actually the breeder score. So we only have this at 3 of the 5 times. Then you have the MGRVI. This is an index which is directly from the RGB camera. Then we have the H, and that's the hue index, and that's an index which is derived from the color transformation. And then the NDLAB, which is also coming from a color transformation. And what is the first thing that is very visible in this slide? So these are the 3 different, let's call it, groups. So here we have tall fescue, here we have diploid perennial ryegrass, and tetraploid perennial ryegrass. And what we see every time here, the time 3 here is always the lowest in the 3 indices. And that makes sense because that's actually the timing right after mowing. So the mowing, the intermediate mowing event, which I referred to earlier, is right before this. So it's very obvious. So it's also nice that it can capture that, but it's obvious that the lowest values are there. And then from this time, you see that that NDLAB and H and MGRVI are growing, at least for the tall fescue individuals. So they're growing again with time, and their biomass is increasing, their greenness is increasing. Now, what we see for the diploid and tetraploid perennial ryegrass is that indeed from time 3 to time 4 there's an increase, and that's visible in every index and for both groups. But we see that from time 4 to time 5, so later on in the drought stress period, also you see that the growth is not taking place anymore, and that's a difference. That's a clear difference between the Lolium perenne, so the perennial ryegrass groups, and the tall fescue group. So this is a very important finding. So we see that the tall fescue group actually, as a whole, performs better. But and then when we look at the different indices, we also see. Therefore, we want to look at time 1 and time 2. Time 1 and time 2 is actually the timings at which drought stress was not very apparent yet. So you can expect the plants there to just continue to grow and increase in biomass, and then you see that indeed the hue value for each of the 3 groups increases from time 1 to time 2. So you see, okay, that's exactly what we expect. So they continue to grow from time 1 to time 2. The same thing is true for the NDLAB. So here you have a growth for each of the species when you look from time 1 to time point 2. Now, when we look at the more commonly used MGRVI, which is directly from which comes directly from the RGB camera, then we don't see this. So we see that the growth is apparently decreasing. See, from time 1 to time 2, in every of the 3 groups, it's decreasing, and that's actually something which we can attribute to the fact that because as grass is growing and increasing in biomass, actually the leaves bend, and because this bending of the leaves, the reflectance is changing. So you have more reflectance when they are bended, and that's a very important feature of the hue and the NDLAB index, is the fact that they are much more performant in this condition. So that's a very important finding for using in our next research, is that actually we should transform our color space from the RGB to the hue saturation value color space, or the CLAP color space. So what I want you to remember from this presentation is that, first of all, adaptation to climate change and to drought requires a systemic approach. So there's not a single golden solution. You have to do it at different levels. And breeding for drought-tolerant varieties is one of these solutions, and it should be used in a bigger perspective. What I also want you to know is, or to remember, is that breeding is very slow. and that the selection should be done properly and should be done better than today. Then progress will be faster. We also want you to remember that RGB images provide very good drought stress indicators, especially when they are transformed to other color spaces like the hue or the CLAP color space. And then also what the final conclusion is that tall fescue shows much better drought tolerance in comparison to Lolium perenne, to perennial ryegrass. And that's all actually also something which has been found earlier, because the tall fescue is known to have a much more extended rooting system. Now, with this, I would like to thank you for your attention. And if you want more information, you can always send me an email or contact me in any way. So thank you very much.